there are hours and hours worth of analysis and exploration of the tracks in the Genome browser that one might want to take a look at. And we're certainly not going to take the time to go through all of that today. But I'll remind you, as I have all along, that any questions you have, you are welcome to ask during the exercises after the break. You're welcome to ask at the end of the day. You're welcome to ask by typing questions into the chat or into the Contact Us link, or setting up a time for a Zoom call with you or others in your group to be able to explore what we're looking at. Let me get started. There may still be some people who are coming back into the group, but my goal today is to kick off what we will be doing for the rest of the day, and just to provide a brief introduction before we break in half an hour's time. Immediately after the break, we'll be coming back to another breakout room where we'll be exploring individual gene models and asking each of you in your groups to explore one or more of the gene models. And we've given you some examples in the exercise here. You'll be assigned to groups, and in your groups, as a group, we'd ask each of you to look at 3 different genes, and those gene numbers are listed here, to explore the reliability and accuracy of the genome annotation. We'll then reconvene in an hour's time to discuss some of the more exciting results that you found. And the overall goal for today is to try to alert everyone to the essentiality in this day and age of being able to explore and understand the vast amount of genomic scale information from RNA sequencing, from proteomics data, from chromatin mark data, from the long list of things that people typed into the chat during Gloria's excellent introduction earlier today. Because we're all used to looking at genes, I'm going to open up a gene. Let's do this. We'll come back to this in just a moment, but I'm going to open up a gene page here. Open up a gene page in Toxoplasma for a change, although I was looking earlier at genes in Fusarium graminearum, in fungi. I've looked at genes in Anopheles or Aedes mosquitoes, or in trypanosomes and Leishmania during the previous exercise. So we're all used to looking at this gene page, and when we look at a gene, we say, aha, this gene, an important phosphoribosyl transferase, essential for the production or for the salvage of purine nucleotides in parasites, and widely used as a selectable marker for studies in Toxoplasma, as those who work on Toxoplasma will know. We can see that this gene reads from left to right on this diagram. It has 1, 2, 3, 4, 5, 6 exons, 5 introns. 5 of those exons are coding sequence. We can see that it starts here and that it stops here. We have a clear picture of what this gene looks like. But as we'll come to know, some of that information is a little bit nuanced. On the gene page, we have a direct link to the Genome browser, and if you click on that link, it will bring you to this Genome browser view that we've already discussed, in which we can see the 6 annotated exons, the introns associated with them, and evidence that comes from deep RNA sequencing, short RNA reads that have been mapped to the DNA sequence, the DNA sequence genome. And when those reads map to this end of this encoding sequence and this end here, that is presumptive evidence of an intron. And just to highlight the spectacular power of the genomic age we're looking at, this particular intron here is supported by 50,000 pieces of information from dozens of experiments and maybe even hundreds of samples. So we have very, very strong evidence that that this gene is indeed transcribed and is spliced as indicated here. The track of or RNA sequence for evidence for introns is available from this list of tracks. Right? So there are hundreds, maybe so several hundred different tracks that are available to explore, and we will only be able to look at a few dozen of those today. We can see the only tracks that are currently active are those for annotated transcripts and the evidence for introns. And because I'm not logged in here, I don't see the other tracks. Oh, here we are. So I can see that there are 600 tracks that I have recently looked at and that I might be interested in coming back to, but we'll learn to try to identify those. As you've already seen, individual tracks can be grabbed and dragged up and down the screen to change the order of those tracks if we're interested. And if there's a track that we're not interested in, we can just delete it. But we don't want to delete that. We're actually interested in the Intron track, which presumably has something to do with gene models. No, maybe not. Maybe I'm looking at the wrong place. I can just use my text search here to type in the term Intron, and I can see a track that I might want to explore for probes that are used for looking at introns from microarray experiments done years ago to this combined track of for RNA-seq evidence of introns. I bring this back to the genome, and here's the track once again. Now, what I can see is that while there are 1, 2, 3, 4, 5 annotated introns and strong evidence for 1, 2, 3, 4, 5 introns, right? So it matches the annotation. My intron track includes 3 subtracks, those that match the annotation. Here's an adjacent gene going in the opposite direction with strong evidence for all 3 introns. But I can also see that there's weaker evidence for these 2 introns and quite strong evidence for an alternative intron that spans these 2. This is a common type of intron polymorphism, an exon skip polymorphism, suggesting that there are transcripts that excise all 5 of these introns, but even more commonly, an intron that completely deletes this intervening exon. Now, I use this example because this gene was the first identified example of an alternatively spliced transcript in toxoplasma from an experiment in my lab a long time ago, 25 years ago. And in that experiment, this is probably the most heavily studied, one of the most heavily studied genes in toxoplasma, almost certainly the most heavily studied transcript. And you can see that in that paper 25 years ago, Robert Donald, who did this work, identified 2 different transcripts by laborious cloning of a dozen or so cDNAs, having identified the original gene. And in that, he mapped 2 different transcripts which differ by the presence or absence of this particular exon and the inclusion or excision of this particular intron. Both of them are annotated as starting at the same point. So we have experimental evidence from years ago. That's not what we want to see. Experimental evidence for years ago for this, and we can see that the exon skip polymorphism here is supported by 35,000 pieces of evidence, as opposed to these 2 alternative introns, which are supported by only, I can just do a mouse over, only about 10,000 lines of evidence. We also see a little bit of evidence for what looks like an intron that has been spliced out in the opposite strand. And when I say a little bit, I mean a very little bit, only 42 reads compared to nearly 1,000 times as many. And similarly here, roughly on 1% of this total. And this is common, that some number of reads in this strand-specific RNA sequencing experiments were mapped to the wrong strand, and so we can probably ignore those changes. But look what else we see. We also see, in addition to these identifiers, we also see weak evidence for other introns that look like they map downstream of this gene beyond what the gene is annotated as, suggesting that there may be rare cases where these genes are spliced to downstream exons that haven't been annotated, and we might want to explore that further. In this upstream gene here, we see evidence for weak evidence for hundreds or 100 introns that suggest there may be an exon skip polymorphism in this gene as well, but at a frequency that's about 2% of the most abundant intron. So this immediately highlights the importance of being able to read RNA-seq data. Do you want to see 2 transcripts annotated for this gene? And I would argue yes. The second transcript is actually more abundant, and we know from experimental evidence in this paper and many others that both of these alternative transcripts are translated and expressed and have different functions. The larger form is associated with membranes, the smaller form is cytosolic. So with strong biological evidence, there's every reason to think that we might want to annotate this transcript. Whereas for this, with an alternative that is there at an abundance of about 2% of the total, I would argue we probably don't want to actually annotate that, but we would like to be aware that it is there. You can imagine that if you are working on the characterization of this hypothetical protein here, and you have evidence for 2 different isoforms from at the protein level or from functional characterization, you'd love to be able to know some molecular data of what that is. And of course, by clicking on these individual genes, we can see in which experiments they're most abundantly observed. This particular one is has been most frequently observed in this stage-specific expression, experiment, but seen at roughly frequency in both tachyzoites and bradyzoites. So this information just shows the presence of the introns, but we would also like to be able to look at the genes that map to the exons. And you already know from the introduction and from the last session how to do that. I'm going to clear the filters and take a look at the transcriptomic data, and I can see there are 343 lines of evidence from dozens of experiments, and I don't really know which one to look at. So I'm going to go back to my analysis here and ask, say, for the most abundantly which experiment has the most evidence? And very clearly, it's from this experiment on feline enterocyte, tachyzoid, and bradyzoite stage transcriptomes. So for the time being, I'm just going to turn on that experiment. I can search for it, looking up and down this list, feline enterocyte, tachyzoid, and bradyzoite stage transcriptomes, or I could type that in here to find the experiment that I'm interested in. And when I do so, I see that this is a strand-specific experiment with 14 forward tracks and 14 reverse tracks, and 4 tracks that are listed as having no data. These are multi-density plots that show all the domains, and they show those that are both unique and non-unique. So I'm going to look at these summary plots at the unique and non-unique data and turn on both of these tracks. Hopefully, there we are. And those will pull up in the background. so we can see what those look like. And I can see that I can see that in this grayscale plot that transcripts are abundant corresponding with these annotated exons, but I don't get a lot of detail. I can see in an overlay plot that the transcripts are most abundant, consistent with these annotated exons. I can see, for example, evidence of this intron exon here. I'm going to use this paintbrush so that we don't lose track of where we are to paint the region I'm looking at. And if I put my mouse over the bar here, I can see the precise organization. So I can see that this exon is less abundant than some of these others. It doesn't look very much less abundant, and it's important to keep in mind that this track is highlighted on a logarithmic scale. So for all of these tracks, I can click at the right-hand end, and I can find information about the track. I can pin it to the top of the track, delete it. I can change the height. Maybe we'd like to change the height of this so we can see things a little bit better. Now we can see things a little better. We can see that these peaks are a little bit lower than those peaks. But most importantly, notice that I'm showing this plot on a logarithmic scale. That's great to be able to identify, to compare across a very wide range of expression, but it means that this short drop here is constitutes a drop from a drop of only about, let's see where this, from about here to here, about 2, 4, 8-fold difference. If I modify this to turn off the log scale and turn on, view this in a linear scale, we can see much more clearly what we're looking at. Evidence for this intron, this one at much lower abundance, exon, this one at much lower abundance, higher abundance, and higher high abundance still, with very sharp breaks corresponding to the introns, and a shaded and a gradual decline at the 5' end and 3' end, indicating that there is no single transcriptional start site or transcriptional stop site. There are transcripts that appear to end across this region, although there are reasons to view that as a possible artifact as well, which we can discuss later on. But certainly we can clearly see that this exon is less abundantly evident, as one would expect from the most dominant intron that spans the that spans the entire region. These peaks are at various heights because this transcript is presumably expressed differentially at different times. Let's just go back to take a look at the transcriptomic data. Yesterday, you saw in the transcription summary data plots that we can observe over a wide range of transcript abundance the expression. Here's the experiment that we're looking at. And if we click on this, we can see that in extra erythrocytic form, in enteric forms that are on the path to sexual differentiation in a cat, this gene is much more abundantly transcribed than it is in the acutely lytic tachyzoite stages, and even less abundantly transcribed in tissue cysts, with a difference of 2 to the 6th fold, 2, 4, 8, 16, 32, 64-fold difference in abundance. Let's take a look at that at the level of the individual tracks. So I'm now going to go back to the same experiment I selected. I'm only interested in unique tracks, but I'm interested in all of those that are present on the forward or reverse strand. And I'm going to first take a look at the acutely lytic tachyzoite stage in both, let's see, the order in which I turn these on is the order at which they'll come up. So I'll look at the forward and reverse strand in tachyzoites, look at the forward and reverse strand in the bradyzoite stage, which must be in here somewhere. There it is. And maybe some of these extra, oops, sorry, Bradyzoite, I turned on, oh, I'm sorry, I'm looking at the wrong strand, the wrong sample. Let's try to find, there's the Bradyzoite stage. And then I'll look at some of the epithelial stages, maybe not all of them, but let's just take a look at the beginning and, shoot, I'm sorry. I'm missing, where were this is the Bradyzoite stage. Turn on the Bradyzoite stage. I could have simply clicked all of these, but I'd like to try to control the order in which they come on. So we'll just look at alternate stages in this cat intestinal lifestyle cycle and come back to see the individual. I'm going to turn off these summary tracks because we're just looking at the individual raw data. And here we can see clear evidence for what we're looking at, again, on a log plot. If we wanted to look at the linear plot, it would look like this. Back to the log plot, linear plot, log plot, and these are expressed at such high level that they've exceeded the max. We can adjust that if we wanted to change the score range or change the height for this graph. But we can see for this gene that that although the annotation suggests that it begins here and ends here, we can see that in fact there is at least one additional 3' transcript addition site, termination site, right? That is quite abundant, but nowhere near as abundant as the annotated region. Should this be annotated as another transcript? Maybe, but in the absence of biological evidence, we'd probably rather not annotate a transcript that is present at only 1% of total message. Now, you'll remember, or you may remember, that in the original annotation of this gene, it was also noted that there was read-through of this initial intron in the non-coding region. And indeed, we can see evidence for that while these two introns are very efficiently excised, there's much more abundant transcription through this first intron. And we can understand why that might make sense in the context of this being non-coding sequence. If transcription begins here and reads all the way through there, it will have no impact on the translational start site, which, let's see, on the translational start site, which is down here within the abundantly transcribed region. But you'll also notice that for all of the work that has gone in, years worth of work in my laboratory and others to characterizing this transcript, there's lots of information we missed. We missed the alternative 3' end because it was only present at 1% of the message. We missed these alternative splice junctions. And remember, we're only looking at splice junctions for which there's strong evidence. If we modify this to look at weak evidence as well, saving this change, you can see there is abundant evidence for all kinds of additional splice junctions in this and other other genes. I'm going to get rid of that because it's not very useful to us today. But there are many thousands of possible gene models. Oops, I forgot to save that. Select tracks, turn that off, and save what I've done to get rid of it. Many thousands of lines of evidence that are consistent. So many thousands of possible additional gene models. Okay, what else was I going to show you? There are, of course, many other data sets that we could take a look at. You might be interested in, for example, transcriptomic data that's associated with oocyst differentiation. And if we look at just the summary plots that are available for that, those will presumably come up. Did I turn those on? No, I did not. Let's turn on these tracks, and now we'll see them loading, and we can see that this housekeeping gene is very poorly expressed in oocyst stages, which are long-lived latent forms, not surprisingly, but we could go in and look at that in more detail. You might be interested in whether there is information on transcriptional start sites, and indeed there is, calculated, determined by analogous methods, but with much greater difficulty to the spliced leader addition sites that you looked at in the last analysis. So let's look for predicted transcriptional start sites, and by going back to them, we can see that the predicted transcriptional start site for this gene here is about here, downstream of what's annotated. And this highlights to us something interesting. You'll remember I pointed out the gradual slope of the beginning of the gene and the end of the gene. And we can see a little bit here. And in retrospect, it is clear that what we thought, or what we interpreted as evidence for read-through transcription, because transcripts were found here in what appeared to be and what seemed to be an intron, was in fact evidence for an alternative promoter site. You can see a pattern similar to there, here, and possibly here, coordinate with this predicted start site. So it's almost certainly true that there is a low-level transcriptional start site here. And of course, if we start transcription within the intron, this intron can't be excised, but the protein coding sequence will be maintained. Now, I'm running up against the end of the time that we have available, and I want to make sure that people have a chance to take a break and come back to explore what we're looking at. But let me just highlight a couple of additional lines of evidence that you might want to take a look at. You might want to take a look at proteomics data, or epigenetic data indicating transcript initiation sites. You might want to look at genetic variation across a wide variety of strains. Lots of different lines of evidence that you might want to take a look at. But let me just close by commenting that once this information is known, that can be incorporated into the annotation in a resource that we won't have a chance to talk about today, in the Apollo Genome browser. And over the coming months, you can look for opportunities for being able to add your own information, as has already been added here. So looking at community annotations from experts in the community, in this case, entered by our expert curator and annotator, Uli Boehm, who some of you will be fortunate enough to encounter in one of the breakout sessions. And what you can see is that this information that we already know about has already been added in the new community annotation, similar to user comments, which will then become part of the official annotation in due course, but which are available to you in the Genome browser shown here. Now, you'll notice that there's a lot of other information that we can explore here. For example, in addition to this gene, we can also see that there are other genes here that haven't been annotated. I mentioned this this intron that corresponds precisely to this much more abundant one on the opposite strand as probably an artifact of the analysis of that. But this intron here does not correspond to the opposite strand. And indeed, we can see that in sexual stage, in parasites that are undergoing sexual differentiation in the cat, there is another transcript on the opposite strand at low abundance that perhaps should be annotated, or perhaps this is a non-coding RNA. Downstream, we can see more sexual stage gene, and another sexual stage gene, which is unequivocally a gene that's stage specific here, was not previously recognized because the annotation was conducted prior to the availability of sexual stage differentiation. And we can see that this gene also has an intron and an exon skip polymorphism. And I won't take the time to go through some of the other points that we might want to make here. Perhaps I will do so at the wrap up at the end of the day. Let me just come back to the assignment for this class. One of the here is a link that if you copy this link will bring up a genome view, not exactly like the genome view that we're looking at here. It'll pull up a genome view that looks like this for an individual gene with multiple annotated introns, some of which are supported, others of which are also supported but not previously annotated. Here, for example, is an annotated intron, but we can see that that there's an exon that's been missed and is far more abundant. Various other gene model predictions. This does not look like what you see on your track here, because this particular track indicating other possible gene models is expanded. I'm going to collapse that so that it's out of the way. Epigenetic marks for H3K9 acetyl marks and K31H4 trimethyl marks and H3K1, H3K4 monomethyl marks. Experiments like the ones that we just looked at, but also some additional experiments as well. So I would encourage you when you come back in your groups to note down the genes that you're interested in exploring. Click on this link to pull up this view, or if you prefer, this link, oops, sorry, this view that we've already been discussing is represented in a long and ugly URL here, which I will store for myself. And during the break, actually, yeah, it's a long URL. It's a very long URL, actually. Here's, let's take a look at. So this information is stored in cookies, and if I open up my chat window, actually, it looks like it's too long a URL to even paste into the chat, but I'll generate a small URL that people can use if they want to look at it in this way. And during our break, during the exercise in the afternoon, you as a group will be looking at these three to come back and identify which one gene of those three from your group you find most interesting and why. And then we will reconvene to hear from each of these groups about what the gene models were that you identified and why you thought that was interesting, and discuss as a group what might be interesting to explore further. So I think with that, I'd better stop and turn it back over to Omar and the other facilitators. Is there anything else that you or the other facilitators think is of burning importance that I haven't yet mentioned?
No, I think just to point out that we will come back and break out into breakouts after the break. So right now, feel free to turn off your camera, go stretch, have a coffee, We'll come back, and as David indicated, we'll have 9 breakout rooms, and each breakout room will look at 3 genes. You can look at more if you want. And the breakout rooms are numbered 1 through 9, so it'll be breakout room 1, 2, 3, 4, 5. So breakout room 1 will be group 1 basically. Okay, with that, see you all in at 1/2 so in about 25 minutes.
I didn't leave time for questions. If people have questions they wanted to ask now and want to stay on, they're welcome to do so. I will stay here, but there will also be plenty of time for questions later on. And I urge everyone to at least get up and stretch and take a break.